The process of DNA replication has many steps, but to fully understand them, we first need to understand what DNA is. DNA, or deoxyribonucleic acid, is a double-stranded molecule that appears in a double helix shape. It is made up of nucleotides with 3 parts: a phosphate group that creates the acid, deoxyribose, which is a sugar without an oxygen, and nitrogenous bases. The way these bases are paired determines our genetics and the variations between them. To duplicate DNA, which is necessary for the reproduction of our cells and occurs in the S phase of the cell cycle, the first step is to unwind the helix. This is done by an enzyme called helicase, which unwinds the 2 pre-existing strands of DNA. It also unzips the DNA, meaning the hydrogen-bonded nitrogenous bases are split apart to allow for the replication process to begin. The next step is done by topoisomerase, which releases the tension of the strand by clipping it. Next, single-strand binding proteins keep the 2 strands of DNA, now referred to as the leading strand and lagging strand, apart. These strands are distinguished by the phosphates connecting to the 3rd carbons or the 5th carbons of the nucleotide's sugars and face in opposing directions. Each strand contains half of the original strand's nitrogenous base pairs. The way these pairs are arranged constitute our DNA makeup and exist in 2 types of combinations: adenine and thymine, as well as cytosine and guanine. In the leading strand, a primase creates a prototype at the 5' end for DNA polymerase to then begin filling in new nucleotides to pair with the old ones. This process shows that DNA replication is a semi-conservative process, meaning that part of the original DNA strand is used in the creation of the new strand. The lagging strand has a more complicated process, as DNA polymerase is unable to go in the 3' to 5' direction and cannot create one solid chain. Therefore, the primase comes back to create a few sections of nucleotide bonding prototypes in the lagging strand in the 5' to 3' direction. Missing strands are then filled in with DNA polymerase. These alternating primer and polymerase sections are called Okazaki fragments and only occur in the lagging strand. The polymerase then converts all of the primase sections into polymerase-created sections before the entire strand is finally sealed with DNA ligase. The process is completed. 2 new strands of DNA have been synthesized.